I'm Suneth Samarasinghe. I'm a PhD student from UNSW Sydney, and I'm also a visiting student at Gowan Institute of Sydney. So, nanopore sequencing is a third generation DNA sequencing technique where the DNA strand is passed through a nano-sized protein pore in a way that it disrupts an ionic current through a membrane. Later, by measuring and analyzing that ionic current through the membrane, we can reduce the nucleotides that pass through the nanopore. So the nature of nanopore sequencing generates long reads, and most importantly, it produces a continuous flow signal data stream. So as we know, epigenetic modifications do not alter the DNA sequence, but it modifies it in various other ways. So here we focus on base modifications, which are very important as they are related to various diseases, including cancer, and also are usually useful in other areas such as tissue identification and age estimation. So nanopore sequencing can now identify these epigenetic modifications, analyzing the same ionic current signal we have. The first step of like almost any DNA analysis is sequencing the sample first, and then at the end of the sequencing is where the downstream analysis is started. So in nanopore context, the issue here is not taking the advantage of continuous output signal stream. So if it took 24 hours for the sequencing, probably will take another 24 hours for the analysis to get a useful insight into the sample. So what we're trying to do here is to perform base modification analysis while sequence is running. On the top, we have the sequencer connect with the sequence acquisition software, generating batch of raw signal files, usually 4,000 records per file. And the challenge here is to make it real-time modification analysis. So we have to process each batch of raw signal files before the next batch comes in, and keep genome-wide live modification records while accumulating the newly created files. On the bottom, we have the solution, and we call it RealFreq. It has three main components. First one is a RealFreq monitor that watches the experiment directory for newly created raw signal files. Then we have the pipeline that converts raw signal files to alignment BAM files with modifications. And most importantly, we have the RealFreq program, which extract those modifications and keep the genome-wide records of modified regions. And also it provides a way to real-time access data in real time, access modifications in real time. So here are the tests we did. First one, we tested RealFreq connected to ONT MinION sequencer connected to laptop. The black color line shows when a new batch is created, and green color line shows where it is processed. You can see that those two lines are staying together means that RealFreq can keep up with the output rate of MinION. We did the same experiment using Promethean single flow cell connected to a desktop computer, and we can see RealFreq can keep up with the output rate of that. So talking about the runtime performance, if we consider the time between two batch raw signal files as 100, the processing part only took 75%, where RealFreq program only took 1% out of it, which is very efficient. If you want to try RealFreq, you can visit this link. It is publicly available on GitHub under MIT license. Also, we have published it in Oxford Bioinformatics. So while testing RealFreq, we noticed our modification computation performing very well, and we decided to bundle it and into a standalone C program we call Minimod, and currently it can be used to view modifications and also compute modification frequencies. So about the accuracy of Minimod, so the major long-read vendors like Nanopore and ONT and PacBio provide similar tools, ModKit from ONT and PBCPG tool from PacBio. But although they are open source, they are distributed under non-standard license. Such license often impose restrictions on commercial users and the data sets we can use using them. But Minimod is open source and vendor independent and distributed on MIT license. So about the accuracy, we compared it with ModKit and PBCPG tools. And the accuracy compared to the gold standard bisulfide methylation frequency results we have, it was like similar to ModKit's output and PBCPG tools output, while reporting many other data points compared to the vendor-specific tools. Also about the runtime, when we ran it on a laptop with DNA datasets, it was 4 times faster on laptop and 1.3 times faster on server compared to the state-of-the-art tools. And especially I want to highlight RNA data here. It was 55 times faster than ModKit and 12 times faster than ModKit on server, which is very impressive. And you can try Minimod, which is also provided in GitHub as open source and publicly available under MIT license. And if you want to read more about it, we are going to publish a preprint pretty soon. And with that, it marks the end of my presentation. Thank you.